Chapter 8, Topic 8.2, Transcription and Translation. The objectives for this topic are: relate the old and new versions of the central dogma, identify important differences between RNA and DNA, draw a picture of the process of transcription, list the 3 types of RNA directly involved in translation, define codon and anticodon, identify in which molecules the promoter, the start codon, and the AMP sites appear, and indicate how you carry out transcription and translation differ from these processes in prokaryotes. In this topic lecture, we're going to be covering RNA and protein synthesis, namely transcription and translation, and then we're going to have some codon reading practice.
Okay, so we have a newly divided cell. It's got fresh new DNA, and now it wants to make proteins. That's what bacteria do. That's what cells do. So what we do is we undergo transcription. That's where we make an RNA copy of a DNA template. So in this diagram here, we get an RNA polymerase, and unlike DNA polymerase, it doesn't need helicase to unwind it. For one thing, it's only unwinding a specific portion of the DNA, just one gene. So it sits down, unwinds the DNA, breaks apart the bases, and it makes an RNA copy of the DNA template. Now, one thing you may have noticed, there's a U here. What's the U doing? RNA doesn't have a T, doesn't have thymine. It does have uracil. DNA doesn't have uracil. Uracil will hold hands with A. Okay, so when we go from DNA to RNA, no T's. Okay, so we make a messenger RNA, an mRNA, from the template. Then once that gets done, the mRNA falls off and we're ready to make some proteins, which is called translation. Okay, to make the protein, we take the mRNA and we attach a ribosome to it. Now, ribosomes are made out of protein and RNA. The RNA is called rRNA, or ribosomal RNA. We also have transfer RNA. Transfer RNAs have amino acids on them, and they carry the amino acids from the cytosol into the ribosome. Okay, so the first thing that happens, and this is called initiation, is the mRNA for methionine, all proteins start with methionine, attaches to its matching 3 nucleotide word. We call it a codon. The part that matches it on a tRNA is called an anticodon. So they match up, A to U, A to U, C to G. Then the large subunit of the ribosome attaches to the small subunit of the ribosome, so that the part of the active site of the ribosome that's called the P site is over the tRNA. Now, ribosomes have 3 active sites. They have an E site, a P site, and an A site. You can think of the A site as being the addition site, the P site being the protein site, and the E site being the exit site. Okay. Another mRNA comes in that matches the next codon. Then what happens, coming down here, the methionine is moved from the first tRNA and is attached to the next amino acid that is on the other RNA, tRNA. Then the ribosome moves down 1 codon on the mRNA. mRNA stays straight, think of it as train tracks, and the ribosome as being a train, and it moves down. The empty tRNA leaves through the E site, goes off, gets another methionine. Then we have the next tRNA that matches the next codon comes in, and the lengthening polypeptide is moved over. Okay, and this keeps happening. Empty tRNAs go out through the E site, full new tRNAs come in through the A site. On the P site, the lengthening chain is moved over. This happens again and again and again. Okay, now when we get to the end, we have stop codons that do not code for an amino acid. There's no tRNAs that match the stop codon. When this happens, the ribosome subunits come apart, the last tRNA comes off of the mRNA, the protein, which has been folding into its active shape as it is made, gets taken off and it goes and does what it's supposed to do, the mRNA can be used to make more proteins. Okay, now I've got another animation for this because this is easier seen when all the parts are moving.
In prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal methionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formal methionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formal methionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of one codon, and the transfer RNA that carried the formal methionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of one codon. As the shift occurs, the two amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
The last thing for this topic will be to practice making a protein from a codon chart, like this one that will probably show up on an exam, hint hint. We are going to start with the DNA, make an mRNA from that, then use the mRNA to figure out what amino acids it codes for. So here's your DNA. TAC, TGA, GGG, ATC. Go back and rewind this if you need to, but do write that DNA sequence down. Using the DNA sequence I've given you, write down what RNA sequence you are going to get. Remember, T goes with A goes with U, G goes with C goes with G. I guess I didn't need to tell you that last part. Anyway, stop the recording while you figure that out. The mRNA sequence you should have gotten is AUG, ACU, CCC, UAG. Now use the RNA sequence and this codon chart to determine the order of the amino acid. Go ahead and stop the video while you do that. I'll walk you through how I would get the amino acid sequence. I look for AUG on the codon chart. I happen to know where it is. It's right here. Right off to the side is MET. That's the abbreviation for the amino acid AUG codes for. It also tells the ribosome, start here. Next I look for ACU. There it is. And it codes for THR. CCC codes for pro. And UAG doesn't code for an amino acid, but it does tell the ribosome to stop, so it's still important. Now you can go ahead and practice this by making up your own DNA strand. We will be practicing this in lecture as well. Now, as a reminder of what the objectives for this topic are, you should be able to relate the old and new versions of the central dogma, identify important differences between RNA and DNA, draw a picture of the process of transcription, List the 3 types of RNA directly involved in translation. Define codon and anticodon. Identify on which molecules the promoter, the start codon, and the A and P sites appear. Indicate how eukaryotic transcription and translation differ from these processes in eukaryotes.